Hello, good morning, and welcome to this Thursday International Plant Breeding Seminar. And we have the honor of having Dr. Juan Acosta from NC State. Juan is a geneticist and tree improvement specialist, and he works here at NC State. As I said, he earned his bachelor degree from the Universidad Nacional in Colombia. His PhD from the University of Florida. He has managed CAMCOR for quite some few years in terms of the breeding and testing program, and works in molecular genetics and molecular and quantitative genetics. Prior to CAMCOR, Juan had worked in Colombia, in Smurfit Kappa, Colombia. And his research interests are in the area of molecular genetics and genomic selection, wood properties, near infrared spectroscopy applications, data analysis, and tree breeding strategies. Juan, it's a pleasure to have you with us, and Juan is going to talk about 40 years of international tree breeding and gene conservation through CAMCOR. I want to stop sharing, Juan, and you can have the floor.
Okay, thanks, Carlos, for the introduction, and I'm really happy to share with the international plant breeding community what we do in terms of tree breeding, which is a little bit different of what we usually do in normal plant breeding. And I'm going to try during my presentation to highlight some differences as well, and make sure that we understand the fundamental differences between normal plant breeding and tree breeding, especially time scale. And I'm also going to talk a little bit about what our program has been doing since its creation, which actually was 42 years ago. And I hope that at the end we'll have opportunity to have a good discussion about how we work in plant breeding. And I'm really happy, as I said again, to present. So let's put a laser pointer. So I work for CAMCOR here at NC State. I'm a house at the Department of Forest and Environmental Resources. We're a small group of people, faculty and staff that have been doing this type of work for the last 42 years. CAMCOR is an industry-driven co-op cooperative that it was funded in 1980, mainly, I mean, as an idea that it was that came from the brain of Dr. Bruce Sobel, which is considered probably the father of forest genetics. At that time, Bruce Sobel was concerned about conservation of genetic important genetics of some species of pine, and mainly in Mexico and Central America. And so when they created this co-op, he hired Dr. Bill Dvorak, which is an emeritus professor at NC State as well, who was the director for 34 years. And they started this alliance with industry members because they wanted to conserve, but not without a purpose. They wanted to conserve species that have commercial importance, but also increase the yield of those species as well. So it's like, we're going to conserve, but we're going to provide some services to industries. And the idea of CAMCOR was, let's try to work together rather than separately. So we have kind of like a motto, which says we share the cost, we share the work, but we also share the benefits. And that's one of the things that I'm going to show probably during my presentation, and I'm going to make sure to emphasize the importance of working together. So we started in 1980 because it was quite an important concern, which is still current, that a lot of species, pine species at that time, but it happens with many species in the world, plants in general, were losing area because of timber harvesting and also conversion of forest to agriculture. Mexico and Central America are kind of like a hotspot for pine diversity. There is like, I mean, it's hard to find out as a specific number how many species of pines are in the world, but there's something between 121 to 126, but there's also a lot of species that still people don't know if they are synonymous or if they're like new species. Now with DNA information, that list changes all the time. But regardless of the number of species in the world, it's like about 40% of them occur in Mesoamerica, like Mexico and Central America. So it's a hotspot, and NC State and Dr. Bruce Oval were concerned about keeping that diversity and use that diversity also with the commercial purposes. So this is just an example of the loss of genetic resources. We have a provenance in Honduras of Pinus caribea, which is a species with a lot of potential, commercial potential. This provenance in our tests, we have found that it grows like about 23% more volume than the mean of that species. And you can see here, for example, in 1988, a picture of the impact that agriculture is having on those. And then you see that same area 4 years after what's going on, right? So in fact, one of the saddest thing is that they remove the best genotypes. You know, for us as foresters, we always want to have trees that grow the most, that have a good volume and good form, and those actually are the first genotypes that are collected or harvested. So losing genetic diversity is important, and that's how CAMCOR is started. Currently, CAMCOR as a co-op is no other than a collection of companies all over the world. This is our current membership. We have 36 organizations that make CAMCOR possible. Some of those organizations are active members. When I talked about active members, I referred to companies that receive genetic material that we collect and conserve and do tests with that genetic material. There are some associate members that are with CAMCOR, but they do not have the capacity of doing testing or breeding. And there are some honorary members, which is mainly governmental organizations in Mexico and Central America, in which we do collection of genetic materials. But if you see here, we are pretty much located in the tropics or the subtropical areas, and mostly also in the southern hemisphere. From the just to show you our members, you can get an idea here of the companies that we have, and also our challenges, because it's a pretty diverse membership. We have organizations in America, Africa, and Southeast Asia. We have those two companies in Southeast Asia, April and Sinarmas. They're extremely huge. They have, each of these companies have over 1 million hectares planted with the eucalyptus and acacias. So very, very big companies. And we have very small companies that have like about 5,000 hectares. So it's, in terms of the size of the companies, is quite of a challenge just to manage them, but we all have a common interest, which is tree breeding, and we work together. But also not only in size, but also in products, and also some of them are vertical integrated, some of them are only greenfield companies. So they just plant trees and sell the trees to the market. Some of them have the whole process. So they start in the nursery and they end up with a paper product or with a construction lumber. We are very strong in the Americas. We have companies in Mexico, Colombia, Guatemala. We have Forest Service is also a member of CAMCOR here in the US. We have an interesting company in Ecuador who grows balsa for highly valuable products. We have a big group of companies in Brazil, Chile, Argentina, and Uruguay. And in Africa, we are located mainly in South Africa, but we also have a couple of companies in Ghana and Sierra Leone, Myro Forestry, and one company one organization in Kefri, in Kenya, which is the Kenya Forest Service. So a very diverse membership, which also makes our work very interesting. So in terms of what they plant, so most of our members plant either eucalyptus or pine. Some of them, like about 55% of our members, plant both eucalyptus and pines. In Southeast Asia, acacia is a very important species. So it's a big proportion of those that is being planted by the members of Southeast Asia. And we have some small members who plant teak, melaina, or balsa, but this is pretty much a good representation of the species or the group of species that are being planted within the CAMCOR cooperative. One interesting fact is our members establish like about 400,000 hectares of forest every year, forest plantations every year, which is very interesting. And to my knowledge, we're probably the biggest organization in the world in terms of hectares managed in terms of forestry. So we work with companies that plant or manage about 5 million hectares, which is an important part of the commercial forestry industry. How do we work? So we have, let's say we have organization in two big components. So we have our CAMCOR staff, which is located at NC State. We direct the program, we talk to our members, we develop breeding strategies, we help them conduct the research and analyze the data for them. We work with them both in CAMCOR programs, but also in company-specific research programs, and we probably show later some examples. But the most important part of what we do is actually our member companies. You remember I talked about active members. So those are members who establish, measure, test. They do the breeding. So we don't do the breeding for many reasons. One, here at Raleigh, the environmental conditions are not the same that our members experience, so it doesn't make sense. Some of those species, even they don't grow here, so the breeding has to be done in their areas. They do provide also the funding. About 90% of the fundings of our program comes from membership dues. So this is very important what our members do. They actually are the heart and backbone of this co-op, and that's what makes this company, our team great, and it's very important for us, our member support. We're a small team here at NC State. Dr. Gary Hodge is the director of our program, and this is our team here in Raleigh. We also have two more members who are located in Guatemala, and what they do mainly is like they do collection of genetic resources in Mexico and Central America. But through this, CAMCOR is bigger than this small team. There's hundreds of professionals across the globe in all the member companies that work with us. And as I said, we share the work, we share the cost, and we share the benefits, and that's kind of like one of my goals, and I hope that at the end this is probably something that you can see and try to replicate, because I think CAMCOR works pretty well. So how we do provide value to our members. So we work in 5 or 6 main areas. We help our members with genetic material for species testing and provenance testing. I'll talk about that in a moment. We work in tree breeding, is fundamental. We also do a lot of research and development. Our work is not only on the tree breeding. We have a lot of new technologies that are also using with our members to help them be a little bit more efficient. We also do special projects that are outside of the CAMCOR umbrella. So like if the company it requires a special quantitative genetic data analysis. We can probably kind of assist them. And the other important thing that we do at CAMCOR is the collaboration and networking. So within the CAMCOR umbrella, there's a lot of interchange of information, genetic materials, and even between species that are competitors. So they are competitors in the market, but they're not competing in getting better genetic materials. So as with many plant breeding, we do tree improvement on what we call the tree breeding cycle. It's just a simple cycle in which we start with some selection. I'm not gonna spend time talking about populations, but you start with a selected individual or a selected population. You take that into orchards in which you're gonna do the breeding, and then you create a new population that go to test, and then after those tests they grow and they're evaluated, you do some selection and you go and start the cycle again and again and again. The way how I see our breeding cycle is more like a sprint. So let's say this is your first generation. So you start when this red dot is, right, this is your current gain or your current phenotype, whatever the trait you're interested on. And the idea is like every time you just move one cycle, you're actually moving up in that sprint and you're increasing the productivity or the variable that you're interested. As I mentioned, for us, it's quite a challenge because we have a lot of diversity. So what we try to manage is like a lot of different sprints in terms of the width of the wire, the length of the wire, you know, how fast those revolutions actually happen, because there's also different interests. We have companies who main interest is solid wood. Some of them, they want to do pulp properties or boxes and packaging. And so we have to deal with a lot of different types of sprints. But that's what we want. We want to kind of like move up whatever app we design, right? And from the selection point of view, we have also fundamental differences with the plant breeding or with most of the plant breeding or crop breeding, is that we have kind of like a lengthy process. So for eucalyptus, we usually select individuals between 3 and 4 years of age, which indicates that probably the total breeding cycle is like about 7 to 8 or 9 years. Pines, we select individuals between 5 and 8 years of age with a breeding cycle of 15 to 20 years. So it's a lengthy process. Genetic diversity is very important because the trees are going to be exposed to a changing environment. So it's quite an interesting work as well. What do we select for in terms of in tree breeding? What we do, probably the most important traits are tree volume. We estimate tree volume by measuring what we call DBH, which is the diameter at breadth height, which is 1.3 meters above the ground. We measure the diameter and we also measure the height. We do also wood properties, because trees are not only volume, but we also want to know how dense they are, what's the percentage of cellulose or lignin, density, many important topics. I'll show a little bit of those later. And also what we call the tree shape. So we want trees with small branches, straight, and also health. So it's an interesting and challenging environment, but we're happy. We don't have time to be bored. In terms of wood characterization, we do a lot of characterization of wood, like with indirect measurements. Some of them could be the structure sampling as well. Sometimes to see what's in the wood of a tree, you have to cut it. But we've been moving into more semi-destructive methods, or something very similar to what we do when we go to the doctor and want to see how our cholesterol levels are, or of triglycerides. So instead of cutting the trees, we're sampling just like a small portion of the wood. And based on that small quantities, we're using a lot of, let's say, new technologies or recent technologies for estimation of important traits. So we've been working with NIR spectroscopy at different levels. So we have very expensive devices, benchtop devices in our lab, but we also have some handheld devices, and we're trying to create new models that work across all those devices. We have many publications. If you're interested in this type of information, you can get in touch with me, and we can discuss that a little bit later. In terms of eucalyptus and pines, we have something that we call our global models, in which we have developed a model for wood chemistry that works within all our members for the most important species that are being planted, and for the traits that are of interest to our members. So for pines, it's very important to understand the lignin and the cellulose, lignin for energy production, cellulose for pulp and paper, and for eucalyptus, also lignin and sugars. And so we have good models for those. We have been also using NIR spectroscopy for some physical properties of wood, like microfiber angle, modulus of elasticity, more structural properties that are important for construction lumber or furniture. And we're also estimating density with a device that is called the IML resistograph, which actually is kind of like a big drill that measures the energy when that drill is passing through the wood. So we spend a lot of our time in not only measuring trees and see how big they are, but also to understanding how's the composition or the characterization of the wood is critical to our members. We have been doing tree breeding for over 42 years now, and with all our members all over the world, we have established more than 1,200 field trials of eucalyptus, pines, melaina, teak, and some other hardwood species. This is just some pictures of tests, but I'll talk a little bit more about later. In terms of genetic resources, I'm only going to talk here about pines and eucalyptus. I excluded what our work in teak and melaina, but we have similar programs, not as big as what we do with pine and eucalyptus, but we have the same type of program going on with those other species. But we have collected seeds in the native ranges from 25 species. So we have evaluated over 360 provenances, and we have selected in the natural population about 8,500 trees. So those, when we select a tree in the wild, in the native area, so that's what we call a family. So we select a tree that looks good. That's when we collect seeds, and that's what we take with our members to tree breeding trials. So currently we have first generation trials in 16 countries. So pretty much all of our members have at least first generation trials. The member who have been with us for more time, they have second generation or third generation trials. So we'll be moving in those loops that I was talking on the spring when I was telling, but this is a lengthy process. So if I'm talking of pines of 15 years of generation, so the ones that are studying the third generation trials are the ones that have been with Camcor for over 30 years. So it's a lengthy process. Some crops you actually have 2-3 breeding cycles in just 1 year. So that's a big difference. In the last 20 years, because our members, so we started as a pine co-op, right? So with the focus on conserving and domestication of genes of those species of Mexico and Central America. But in the last 20 years, or more than 20 years ago, in 1996, our members decided that it was important for Camcor to work also in eucalyptus. Is probably the genus that is most widely planted in the world, and now we have like about 23 species of eucalyptus for which we have tests or collection. So you can see here that we have many species of eucalyptus in which we're doing breeding programs. So now you understand when I was talking about those different springs, those different tree breeding programs. So it's quite a challenge just like to work with all of us and try to move forward in those breeding cycles. One of the interesting things of CAMCOR for our members is that we provide diverse species for diverse sites, or for diverse environment. As I said, 25 species of pines, 23 species of eucalyptus. The idea is we see sometimes CAMCOR kind of like a health insurance or like a insurance for the company, right? So if something happens, I'll show you some examples soon. It's important to have a replacement species. So this is, for example, one idea of having a alternative species in the southern Brazil or the northern Argentina, say in the province of Misiones, or in Paraná and Santa Catarina in Brazil. The most planted species is Pinus taeda, right? It's been planted and improved for over 50 years, and in that part of the world, the Pinus taeda it actually yields the highest productivity. We're talking about in forestry, productivity is measured as cubic meters per hectare per year. And this is a really good value. So I'm talking about 35 to 45 cubic meters per hectare per year. Pinus taeda is native of Southeast US, right? It's actually, North Carolina is actually one of the natural populations. It goes from Texas all the way, along the East Coast to Maryland. And here the productivity is not as high as it is in Brazil and Argentina. But in that area, we have had alternative species for unimproved sources like Pinus maximinoi, which is a species with a lot of potential in the tropics, but as well in Argentina and Brazil, that unimproved at 8 years actually can yield more than between 50 and 60% more wood than Pinus taeda. So if something happens due to climate change or a new pest and disease that attacks taeda in Brazil and Argentina, so there are some alternative species that the companies can use to pretty much to remain alive. Okay, some other species here that we can see, for example, in eucalyptus is Eucalyptus dorrigoensis, which is a species that is very tolerant to cold. It has a lot of potential in the southern hemisphere, mainly in Argentina, Brazil, Uruguay, and South Africa. And it grows as well as Eucalyptus benthami, for example, which has a lot of restrictions in terms of genetic diversity. It's because it's from a very small region in Australia. So having alternative species can also help our members. And what we have learned from working with those new commercial species, as well as doing breeding on the main species, because we help our members work with loblolly pine or Tecunumani as a main species, for example, in South Africa and in Colombia. We have been also working and learning in the last 20 years, probably half of our life as a co-op has been working with hybrid breeding. And this is what I'm going to spend probably most of my time. I'm going to talk about hybrid breeding and why we do it. So first of all, hybrids are fun. That's what I want to show you. And hopefully at the end, you'll see that I might be a little bit right. But even though it's fun, it's hard. And when we talked about breeding, it's probably a little bit different from some crop breeding. We actually talked about interspecific hybrids. So we're actually talking about two species that are not the same, okay? And so because of that, there are biological barriers that avoid crossing. There's also logistics challenges because companies usually have to maintain 2 breeding programs. And there's also challenges in terms of operational mass production. Breeding hybrids or hybrid fruits or cones in pines, they don't yield as many seeds as a pure species. And so we have to work with vegetative propagation or second generation orchards for open pollination. So it's a little bit challenging, it's hard. But so if it is hard, so why do we work with hybrids, right? So one thing is like different species have different advantages. So some species grow better, they are adapted to certain conditions, some have some disease tolerances, some have better wood qualities, some might have more lignin or more cellulose, which is probably interesting for a company who produce energy, or for a company that is interested in pulp and paper. So when you have different species, you want to put them only in a single package, right? And that's what we try to do with pine hybrids. So this is and at the end, what we're doing is we're actually trying to use heterosis, right? So you can see here in this picture in Zimbabwe, like how a pure species of Elioti grows, right? But then just look at the next row, and then you can see that this is a hybrid between Pinus elioti and Ocarpa. So you can see that heterosis usually is present in growth rates. You can see what you can gain just by mixing 2 species, okay? In terms of diseases, in the last 15 to 20 years, we have seen a lot of increase in diseases affecting forestry all over our members. So there are a few of those species that I'm showing you here, how important they are. Some of them in blue, the others are important for pines. In green, the others are important for eucalyptus. That they translate into productivity losses, and which it'll be at the end economical losses. I'll show you how we do breeding and how hybrid breeding actually help us to do this. The other importance or why we think hybrids are important, because we all know climate change is a reality, and it's a big challenge that we're actually facing. And with trees, you actually have to plan today what you're going to harvest in 20 years or 8 years if you're working eucalyptus, but it's 22 years if you're working with pines. So you have to have a good genetic diversity so that your genetic material can adapt to changing environments. In terms of pine hybrids, in 2020, after spending, let's say, the first 20 years of our program on single species breeding, we started to develop our first CAMCOR pine hybrid program, in which we work with all members, and we have established like about 90 trials with our companies, within our members. So it's a big network of tests which are providing information that I will share with you. So those are the pine species for which we have made hybrids. So this is some just pictures of the test, but you can see they're mainly distributed in Latin America or in Africa. Our companies from Asia, they're kind of like they joined our program in the last 5 years, so we don't have lots of pine hybrids with them or eucalyptus hybrid with them. But we have a good number of tests located all over the world, and this is what we have learned, right? Between 2000 and 2015, when all those breeds were created and established and developed, we have seen that pretty much in all the environment, there's always a hybrid that is equal or even exceeds the commercial species. So those are 2 graphs of part of our analysis at 8 years for volume in Argentina and Brazil, when I was telling that Taeda is one of the most productive species. But then you can see that the hybrid with Caribbean and Peg or the Ellioti by Caribbean, Carpa, they're by far superior than what you get there operationally. And in terms of, this is the module of elasticity, which is a structural trait for wood. You can see that some hybrids are as good as the best species here, which is Tecnomani. So hybrids, we always found individuals or hybrids that are better than the commercial species. In terms of disease potentials, in 1988 in ChemCorp and with the Forest Service here and North Carolina in Asheville, we screened different pine species, and for a very important disease, which is called pitch canker, which causes like dieback of the seedlings in the nursery, and it actually can translate it into the field, which is very economically important. And then if you see here on the y-axis, we have survival, and here on the x-axis, we have different species. You can see that there are some species of hybrids, some pure species for which survival is pretty high. In this case, we have Tecnomani and Ocarpa and Haliscana, some species that are very resistant to Fusarium, but you see Patula, Radiata, some of those species that are very important commercially, they actually have very low survival. So when this disease actually gets into the plantations, the operational plantations, it has a very important economical damage. So this is, for example, for one of our companies in South Africa, in which pinch canker from the nursery is being carried out into the plantation, and you can see how the survival actually decreases. And you don't want to have plantations for 20 years with an initial survival of less than 70%, let's say 60-62%. So you see how the effect of one disease can actually change the silvicultural practices. So that same company, they did full seed process between Patula and Tecnomani because of our results of the disease susceptibility and tolerance on our screening. And you can see here Patula and Radiata, the two main species for them. They actually have a high percentage of incidence. Tecnomani as a pure species have a very low percentage, but then the hybrid is actually closer to Tecnomani and Patula. This has been very important because that's probably now the most important hybrid planted in South Africa, which is the hybrid between Patula and Tecnomani. And this is one picture in one of those stands that we were visiting. And at 5 years, we can see, I mean, it's not that clear if you look at higher detail, but I'm just going to kind of like translate what the numbers are. So in yellow, you can see the hybrid. In blue, you can see the pure species, Patula, which was the commercial plantation in that area before. So you can see, or the bars indicate that in terms of survival at 5 years, you have a 90% survival of the hybrid, whereas you have a 70% survival of the hybrid. You also have better growth. You have like about 80% more DBH, more diameter of those trees, also 12% more height and about 34% more volume. So you are producing better trees with more volume. And as a bonus, when we study the wood properties, we see that there are higher density, better modulus of elasticity, and better or higher modulus of rupture. So it's a new hybrid species, or a new hybrid combination that provides more growth, better adaptation and survival, and also better wood. Some other pictures. This is in Argentina and Uruguay. You can 5 years of age for a hybrid between Greggii and Tecnomani. This is quite great to have trees at 5 years that are this size. And same here with Greggii for Tecnomani in Brazil. See 5 years of age in Santa Catarina in Brazil and the state of Paraná. You can also see very promising trees or promising hybrids. So because of all these results that we've been doing with our members, we decided to start working on what we call the collaborative full sieve breeding. So when we started our pine hybrid program, what we did was just tests. We just did breeding without targeting specific families. We just wanted to see the behavior of the species. And everything that I have shown you so far is based on just general combining abilities. We're just selecting some females and some males from those species and just making crosses. Now we're moving with our members into a targeted full sieve breeding in which we're actually selecting the best individuals for species 1 and the best individuals for species 2 to start making crosses. This is a very lengthy and expensive process, and when we do it is we work as a team. So like a company 1 does one hybrid, the company 2 does the other hybrid, and so at the end, all companies produce 1 or 2 hybrids, but all of them test 20 or 15 different hybrids. Now we're working in those 3 main programs. One is Patulan Tecunumani because of his important in South Africa or in Southern Africa. We're working with Tecunumani and Greki, which is quite important in Latin America. And we're working with Caribbean and Tecunumani, which is a hybrid that's important all over. In terms of how we're working, just to give you a couple of examples, how we do that work. So for example, Gregi by Tecunumani, what we're doing is some members in which they have the species 1, in this case, let's say it's Gregi, they collect the pollen and it's being sent to our group, our lab here at NC State. We collect the pollen, we do germination tests, and then we send to another member who's gonna do the crossing. So in this case, collections of Greki as a parent were made in Brazil and Argentina. And they were sent to Colombia, which they have a big orchard of Pinus tecunumani in which all the crosses are being made. In this case, we're targeting 300 full sieve families with 100 parents, 50 parents of Greki and 25 parents of 2 subspecies of Tecunumani. We have a subspecies high elevation and low elevation. Okay. The other hybrid we're currently working now is Caribbean by Tech. Again, Taeda is, this is a picture in Argentina where the main species is Taeda, and you can see here that the top of those trees is like around here where I'm actually placing my pointer. And that's the commercial species, but then you can see the row behind when you have the hybrid, the big advantage that it might represent for our members. And this is values of volume, and you can see that the hybrid by far surpasses the volume produced by Greki. I'm sorry, by the main species, in this case, Taeda. We have also a collaborative project in which members all over the world are actually collecting pollen of Tecunumani and are sending that pollen to our lab, and we are shipping that pollen to South Africa when our members have the best genetic material. Once that pollen is being produced, then we receive the seed again, and then we send it to our members. We're doing something very similar with hybrids for eucalyptus. We have been doing this in 2 phases. The first phase, 12 companies produced 12 hybrids, and were companies from South Africa, Colombia, Uruguay, Brazil, and Chile. That is a process that started like 6 or 7 years ago. Again, this is a lengthy process, and in 2020 and 2021, the tests have been established in the field. And now, a few years ago, 3 or 4 years ago, we also started the second phase on eucalyptus, in which more companies are participating and more hybrids. Just to show you the hybrids for which we're focusing our breeding effort. This is the first the first phase, only 12 hybrids. And in 2020, 24 tails of those hybrids are being established in South Africa and 3 in Latin America, South America. And the phase 2, we're still doing crossing because there's a lot of things that you need to align, right? So you have to, I mean, in terms of biology, you have to make sure that you can collect the pollen, that the pollen germinates, and then you have to isolate the flower, make the process. It's challenging, but it's even harder if you do it by yourself. So we do share a lot of the workload, and we also share our benefits. In terms of our experience, in terms of hybrid breeding, we say it's hard, but it's very, very important, and the hard work is going to be compensated. In our experience, in pine hybrids, we started from understanding the hybrid. Now we're working a targeted scheme. So we're actually working with families that we expect are gonna be better families than just random families. But we also have seen a lot of issues with incompatibility, frost, and hail. So there's a lot of operational challenges. In eucalyptus, we have done the 2 phases that I just mentioned, but we also have an important differences between eucalyptus and pines is that the eucalyptus has the capacity of sprouting and rooting. So when you select a hybrid, you can actually clone those and propagate that vegetatively. So it's a little bit easier to work with. So we expect even higher gains. And as I've been telling you, when we work together, we share the work, we share the benefits, and it's hard. And we believe that if you want to start working on hybrids with trees, you have to start now. And there is a Chinese proverb that says that the best time to plant a tree is 20 years ago, and the second best time is now. But in terms of breed a hybrid, it also was 20 years ago. So we have to start now, and that's pretty important. My last 2 slides are related about some other topic that I want to mention. We have been working, ChemCore has been part of the Conifer SNP Consortium. Pretty much everything I showed you before is based on traditional breeding, but we're also working a lot now into including genomic tools into our breeding programs. So there is a Conifer SNP Consortium that is led by FICRED at the university here at NC State. And our group, in conjunction with the Forest Molecular Genetic Group at the University of Pretoria, develop the tropical SNP chip. We use sequencing data, and now we have a 50,000 SNP chip that is available for the community and that we're actually using now for molecular system breeding. What we want to do with those techniques, or what we're trying to do now, which is a lengthy process again, and we're just starting working with, is kind of like change the shape of those springs, right? So we're still going to have to move generation after generation, but what we want to do is kind of like start doing smaller cycles. So can we use molecular markers as well to reduce the length of that breeding program? Can we do early selections? So that's pretty much how we anticipate the molecular tools will help us moving forward. There's we have 4 projects currently with our members in which we're using genome-wide selection to improve full seed family forestry. And the idea is that we actually are focusing on doing genomic selection only on the best families of your breeding population. So let's say this is your distribution in terms of volume. So that if that's the overall distribution, you can also find out that there are some family distribution or different treatments within this. What you want to do is kind of like focus on those best families and see if we can target it or create genome-wide selection models within each family so that we can increase the gain within each of those families. So we know those families are good, but what if based on molecular markers, we can develop a good model so that we can select the best individuals that are going to be deployed into the operation. So we've been working that with the companies in Brazil and South Africa, and we're going to start pretty soon with another company in Colombia. But as I show you, those genetic resources for pines are being developed in the last 4 years or so. So this is, even though the technology has been always there, is not as highly adopted as it is with some other plant species or with humans. So it's a little bit more challenging. So that's all I wanted to say. So ChemCore has been changing a little bit from gene conservation and domestication to hybrid varieties. So we do believe that from the forestry perspective, hybrids is the way to go. We have plenty of data, and we know how important is this. And thank you, Carlos. I think we have some time for some questions.
Thank you, Juan. Thank you so much for such a nice presentations. Yeah, we have a few questions already. And one of them is Emmanuel is asking, what are the opportunities for students to get involved with ChemCore level?
There are many and too little at the same time. So let me explain what. We are a program that is funded 95% for our members. So we do have lots of grad students, but they are mainly students from our members. So
also by your members. Okay.
Yeah. So currently we have 5 students, and 4 of them are actually employed by the companies who pay. Who support the program. We do have students that are not part, or we have those, but we might have to find out external funding. So if it's an international, and I'm saying this because it had happened, there's a lot of possibilities for international students to get funded in their countries. So if you have funding available and you want to work with us, let us know. We'll be happy to have you.
Okay, good. Just for your information, for the audience information, Brandon has posted a link to the ChemCore publications, which may be useful for some of you if you want to get into more detail. Question from Russian. Could you speak of what extent we can expect to improve gains through hybrid breeding? I think you talk about this at the end through several generations. I think you show that at the end.
Yeah,
but I don't know if you want to elaborate.
Yeah, no, that's a great question. So there are many gains, some of them not quantifiable immediately, right? First one is survival. I mean, if I show you in South Africa, if it's not because there is a hybrid between Pinus tecumani and Pinus patula, they shouldn't be a Pinus plantation in South Africa. All patula plantation, which they were being planted for more than 100 years, all those plantations are now pretty much gone, right? New plantation are hybrid. So just keeping productivity is very important. But I also show a slide in which you see that you have like a 35% more volume, you have better wood properties, you have better survival. So yeah, I mean, it's definitely an advantage.
So question from Sadhu, is distant hybridization possible in tree breeding? If yes, what are the techniques involved? And I want to add, because I'm very interested about this conversation about hybrid, how is seed production?
Yeah, that's the big challenge. I mean, because nature has been evolved into maintaining the species. We're actually playing against nature. When you're working against nature, you're going to have a lot of issues, right? Yep. So first, there are some species for which, even if you have pollen and you have a receptive female, so the size of the grain, the pollen grain, sometimes is too big that it doesn't go into, it doesn't do the germination. So there's a lot of physical barriers. And so what we have done, if we find that case, we switch the species. We actually try to talk, instead of being across 8 times B, we do it B times 8, right? In terms of seed production, is pretty low. I think I mentioned that. So when you have a cross between a full seed family of Taeda, you can get 40, 50 seeds per capsule, per fruit. When you do it in a hybrid, you can get 5 or 7 of those. And so that's a little bit tricky. So what we're doing now is that we spend a lot of time into doing that breeding effort, but then when we do the deployment, we actually do vegetative propagation. So we use one seed and we multiply that seed in hundreds of thousands so we can actually test it. But it's a lengthy process, but it's rewarding. It is, and sometimes it's just the only way to go.
Okay, so a question is not in the chat. My question I have written here is there possible to do breeding within the hybrids themselves, not just the parents? Okay.
Yes, and that's what we're doing with. I didn't mention that, but that's what we're doing with some companies, specifically in South Africa, because there are two things. You can do hybrids maintaining the two lines, right? Species A and species B. So you have like two springs in which you're doing breeding, and then you just join them in a hybrid event, and then you move forward. But also there are some companies in which they say, no, I'm going to have a hybrid population, and then I'm going to do selection within those hybrids and start moving forward on a hybrid population already. Mm-hmm. Because of segregation, you're probably going to have more variation in a hybrid population. But once we have good genomic selection models, we have good molecular information or some market assistance tools, we could probably filter out that variation and select the individuals that are going to move forward. But we're also doing, we're moving also the hybrid generation.
Elvia is asking if some of this hybridization work has been attempted in Peru.
Unfortunately, we do not have members in Peru. We in South America, we have members in some other different countries.
But it will be potentially adaptable if you have them.
Yes, it can be.
Okay. Emmanuel is also asking if you have any affiliates in Nigeria.
No, unfortunately not. Okay. We have been in many countries in Africa. Currently, we are only in Kenya, Ghana, Sierra Leone, and South Africa.
Okay. And another question from Roshan, interested in to know what your strategy is for reconciling breeding zones for your new hybrids.
Well, that's a very interesting breeding zones or breeding sources.
Yeah, I don't know if zones or sources, Roshan.
If it is zones, I'm actually going to talk about zones, and if it's sources, I can also tell about sources.
Yeah, zones. He clarifies his zones. Yes.
Okay. So in terms of zones, we usually, and that's why it's important a group work, right? So breeding zone is actually when you can have the species flowering. That's very important. So for example, Pinus caribea, it grows a lot. It's a very interesting, but it has very limited environmental conditions in which it actually flowers. So our Pinus caribea by Tecnumani hybrid program is actually focused about doing all the breeding in South Africa and in just a very small place in South Africa, which is called Zululand. That's where Pinus caribea actually grows. So that's our breeding zone. But the collection of the male, the pollen actually comes from all over the world. So companies in Colombia, Brazil, South Africa, I guess Chile, they are actually contributing pollen to do the breeding over there, right? In terms of zoning, it also depends on how the hybrid performs. And that's why we did this network of 90 different tests just to identify the best hybrid and also where they will grow well. So it's a lot of testing and evaluation just to make sure that you get what you want.
Okay. Well, thank you so much, Juan. I think we are out of time. I just want to announce the next seminar, which is going to be next Thursday. Hold on a second. Well, first of all, thanks for the attendance. I mean, it's a very good seminar. It's going to be, the recordings are going to be shared pretty soon in our website, and you will receive a communication. Thanks to Brandon and Tiago for all the help you guys provide in organizing these events. Next seminar, as I said, is next Thursday, the 11th at the same time. Elcio is going to talk about rice breeding in Brazil, a very successful story. I think it's an extremely interesting topic, and I will invite all of you to attend. Thank you so much, everybody. You guys have an excellent week, excellent weekend ahead. Thanks, Juan, for your presentation. It's been a pleasure. And
My pleasure.
okay, thank you so much. Bye, guys.
Bye.
Bye-bye.